Welcome to the Bland Lab. We have a lab that focuses on behavioral neuroscience. We're interested in a number of sort of different topics or strands, but sort of overall, we're interested in the intersection of social interactions as well as social stressors such as social isolation and how that affects an individual's response to drugs of abuse. Not all of our experiments include the drugs of abuse aspect, and so some of our experiments really just focus on what social isolation does during the critical developmental period of adolescence. So what does that do to the brain? What does that do to subsequent behavioral interactions with other rats and so on? So we do study rats. We use rat models because we then can look into the brain and look at changes in the brain either after or even during some of these experiences. Some of the techniques we use allow us to look at different proteins in the brain, and some of the students will explain how they do that and what kinds of things they can actually see in the brains of rats. And some of our experiments actually can be done in awake, freely behaving animals, and we can actually assess neurotransmitter levels, like dopamine and serotonin, for example, which are known to be affected by drugs of abuse as well as stressors.
So Jesse's going to be using a PBS solution, phosphate buffer saline solution. We use this a lot for washing the different tissues in order to do for IHC and other methods. She's combining 100 milliliters of PB, the phosphate buffer, 900 milliliters of double distilled H2O, and 9 grams of sodium chloride. And then we leave it on the mixer with the magnet at the bottom mixing everything up. And this gets stored in the fridge for when we need it. I'll be introducing IHC, immunohistochemistry. We start, the purpose is to tag things like proteins with antibodies, and we tag the antibodies with fluorescence in order for us to look at it at a confocal microscope. We start out by washing the tissue in a phosphate buffer solution on the shaker, and then we move on to a blocking solution, which is composed partly of, this is normal goat serum, I just like the name of that. And then next we move on to the primary. The primary for this one is CFOS, and the concentration is 1 in 10,000. So when that means that there's a little bit of faith involved when you're trying to pipe at 1 microliter. There's nothing to see. It's interesting. Next we move on to the secondary. This is the fluorescence part. So this has to be done at night, or it has to be done with the lights off, please. So we shut everything off and we make the solution. And we put it on the shaker overnight with a box over it. The next day we come in, usually as a group, and we have to do 4 more washes, and that's when we have to take all the tissue in those tiny thin slices, and we have to put them onto these slides in the dark. That's actually a process. There's a little bit of swearing that goes on. That happens. After that gets done, we put a couple drops of this right here that stains the cell bodies. We cover slip it, and we move up to the confocal as fast as we can, because the payoff is really good. It's interesting. The colors are right on the screen. There's dimension to it. It's a lot for what I was explaining earlier with the IHC, where we need to wash the tissue in between different procedures for the original to add the blocking solution, and also between the primary and the secondary, so that we have less background when we go look at the confocal microscope.
Hello there. I'm Raleigh. I'm a sophomore at the University of Colorado, Denver, working in the Bland Lab, and I'm going to show you cressyl violet staining today. So cressyl violet is a compound that is used to stain Nissl bodies within rat brain tissue. A Nissl body is an aggregate of ribosomes, which are the site of protein synthesis. So we use this to better visualize the samples when using light microscopy, specifically to determine probe placement for microdialysis testing. So when we're looking at crescent violet staining, we use a series of ethanol baths. So we prepare the sample, put it in an ethanol bath, which clears it out, and then we'll stain it in a crescent violet solution. And then we'll wash it multiple times to make sure all the dye is out. And it'll result in a slide with very well stained rat brain specimens. And then we can look at the slide under light microscopy and better visualize the difference versus an unstained sample. So that was just a quick once-over on crescent violet. It's used in a lot of different neuroscience labs, as well as botany. It's used in gram staining of bacteria and some other subjects. So it's a pretty wide-ranged procedure. But if you're pursuing a career in neuroscience, odds are you're going to have to do crescent violet staining at some point.
So the tissue that we're looking at here is from a rat brain, and specifically we're focused on the prefrontal cortex, which is an area that's associated with higher order processing. So it's where a lot of decision making is done, where rats sense of smell comes from as well, which we know is a huge communication piece. And so just to get a sense of the scale here, we're probably looking each little green dot here is about 1/100th of a millimeter. So the protein that we stain for in this tissue is called ARC, and ARC is the product of immediate early gene expression. It's a type of protein that's expressed after neuronal activation, and so we can quantify this expression and decide what the neurological effects of social isolation were in those animals.
Experiments that we do involve the assessment of neurotransmitters in the brain, like dopamine, which is associated with reward, and serotonin, which is associated with stress. And those two neurotransmitters can kind of counteract or act against each other. So we like to look at both of them in the same animals. And we do that using this technique called the HPLC. So we get the samples from the microdialysis experiments that Jessica will tell you about. And we actually start out with very tiny samples. So I don't know if you can see that, but this is about 40 microliters of sample. And we can get multiple samples for each subject, though, so we can sort of look at changes across time in these neurotransmitters. But this is just to give you an idea of how very tiny it is, and we get very, very small amounts of these neurotransmitters, which we can then assess using this equipment, which is actually something that's typically thought of as equipment that's used by chemists, but even psychologists can use it, like me. So now I'm just taking this tiny sample and loading it up into this vial, because the nice thing about this technique is that it's all automated. Things have really changed a lot since I was a graduate student, and I had to do a lot of this by hand. But as you can see, we have now very shiny, nice new equipment that helps us do this. So this is an auto sampler. I can take the samples and load them up in here, just like that. And what this will do is it'll inject the sample into the HPLC, which I won't go into the nuts and bolts about how it works because it would take too long, but we can essentially use analytical chemistry to both separate and detect our neurotransmitters of interest. And the whole thing is driven by our software. And so here I actually have an example of what you might see in an actual microdialysis sample. And here you can see this nice peak, that's dopamine. So this shows us the amount of dopamine that's in the nucleus accumbens of the rat. So this is a region that's very associated with reward, but can also be affected by stress. And this tiny little peak here is serotonin, poor little serotonin. So notice there's a lot more dopamine in this brain region than there is serotonin. So we have to have very, very precise tools to be able to detect these tiny amounts. So these, even though this peak looks nice and big, this is on the order of picomolar amounts, which is, you know, a very, very tiny amount of a neurotransmitter. And we can run multiple samples per animal, as I said before, and look at changes across time if they're exposed to a drug like cocaine, or if they're exposed to another animal. So we can look at changes in these neurotransmitters as they're engaging in social interaction, and we can look at the two together. Because one of the things I'm very interested in is how does being with a peer or a playmate change the effects of the drug? Because in terms of clinical relevance, a lot of labs just, you know, give drugs to their subjects and just throw them in a box. But in the real world, most people, especially adolescents, which is the group we're interested in, do their drugs, especially at first, with a peer group of friends. And we think that might be a very important variable in what may make individuals more vulnerable to addiction. If that social aspect is adding some additional rewarding aspect. And so we can actually look at these neurotransmitters and look at relationships with addictive behavior and things like that. For the microdialysis experiments, we surgically can implant a guide cannula like this into specific regions of the brain, and we can be very specific in our placements of those, so we know exactly where we are. We're interested in regions, as I said before, like the nucleus accumbens, the prefrontal cortex, and other regions that are involved in things like reward, social behavior, emotional regulation, and like that. So this guide cannula is surgically implanted into the brain, and then we allow a week or two for recovery. Recovery actually happens pretty fast in the subjects. Once it's time to do the experiment, we can then implant this, easy for me to say, right? We can then insert this this probe into the guide cannula. This one's not wanting to go in very well. And you can see a little bit of it comes out the other side, and it's covered with a semi-permeable membrane. And once we start the experiment, we can actually perfuse a salt solution, so it's artificial cerebrospinal fluid that's designed to mimic the fluid that's in your brain. So it flows through all this tubing and it comes out the other side, and anything that's in the brain that's small enough to go through that semi-permeable membrane will do so. It crosses down its concentration gradient, it can come out the other side, and we can collect those tiny samples with very tiny bits of neurotransmitters in them and run the HPLC to assess the levels of neurotransmitters.